we are going to talk about advances in genetics. So, first off, you should know that the Human Genome Project is the project that is used to identify the DNA sequence of every gene in the human genome. It's never going to be 100% done, but we know what 99.9% of the genes inside the human genome do, and basically most of the time how to manipulate them. So different genes code for different things. Sometimes we can turn them off, sometimes we can turn them on, sometimes we can erase them completely and snip them out, sometimes we can add more genes in. Those are more recent developments in the human genome, but those are things that we can do. Your genome is just all the DNA in a normal body cell of you. Okay, of any organism. All the DNA in one cell of that organism is called the genome of that organism. Genetic engineering, this is genes from organisms that are transferred into the DNA of another organism. So basically, when we have genetic engineering, we're playing with the genes that exist inside of your cells. Genetic engineering can be an example of bacteria that produces insulin, or cows that produce milk with blood clotting protein for hemophiliacs. So that way, even though the people who are hemophiliacs can't produce the protein, they can drink milk with the protein and therefore still get it. It's a treatment, so it is a genetic engineering. So one way of genetic engineering, and this is one that is very common and has been around forever, since Mendel was around in the 17th century, and this is selective breeding. Selective breeding is a process where we select organisms with the desired traits to be the parents of the next generation. Now we have two different traits. We can either inbreed or cross individuals who have two similar characteristics, or if we don't want just a single trait that both people have, if we'd like a combination of what two people have, we can cross two genetically different individuals in the hopes that the offspring will have the best traits from both parents, and kind of make a hybrid. So again, inbreeding is crossing two similar individuals, hybridization is crossing two different individuals. Cloning, you guys got a little taste of this today in class, but cloning is just when you take an organism and make an exact replica of the genes from which it was produced. So basically you have a mom and a child and they are genetically identical. Kind of like when cells go through mitosis, except these are full organisms, like a sheep, like Dolly the sheep was a clone of her mother. Gene therapy. This involves inserting copies of a gene directly into a person's cell. So basically, if they are missing a gene or the gene is not functioning, incorporating that gene or turning that gene on inside people's cells. And this is a way we can work on people who already have a genetic problem but who are already born. This is a way we do that. Okay, so those are the big ones. Again, today we talked about some other ones. We talked about GMOs. You're going to research those more. Stem cells, designer babies, all that kind of stuff. So you're going to research all that stuff more in the next couple weeks. Look around the website and you will find some different, or my website I should say, and you will find some different videos on each of those things we talked about today. All right, I hope that makes sense. Thank you.